Right, onto amino acids. So if you remember from the first lecture, I gave you a preview of these, and that we'll go through them again in a little more detail. That your amino acids we're going to use are specifically called alpha amino acids. And what that means is we look at the carbon numbering system in any molecule, any organic molecule. We find the highest priority carbon, and that's going to get number 1. So in the case of our amino acids, the highest priority carbon, our functional group, is our carboxyl group. So carbon with the 3 oxygens attached to it, or 3 oxygen bonds to it, will be our carbon number 1. And of course we go down the chain labeling it 2, 3, 4, 5 until we get to the end of the longest chain. So in that case, this carbon would be number 1, where the carboxyl is. Carbon number 2 is where the amino group's attached. And then the R group, if it has 1 or more carbons on it, would be 3, 4, 5, etc. Okay. Alternative naming system is using the Greek letters, where the carbonyl carbon, the first carbon, doesn't get a letter. It's just called the carbonyl carbon, or carboxycarbon. And the second carbon, being the carbon number 2, or alpha carbon. Carbon number 3 would be beta, carbon number 4, gamma, and so forth down the Greek alphabet. Right? So all of our amino acids have the amino group on carbon number 2. So they're could be called 2 amino acids or alpha amino acids, because that's the position of the amino group on an acid, or carboxylic acid. So they're all alpha amino acids for our 20 we're going to use, and the R group is what varies among them. Some examples at the bottom. You see we're drawing these all the same way. The carboxyl group here, the amino group attached to the carbon number 2, and then valine has a beta carbon, so carbonyl carbon, alpha carbon, beta carbon. The beta carbon is branched, right? There's 2 methyl groups on it. So one of these is called the delta carbon, the other one's called the delta 2 carbon. There's 2 deltas. Okay? For cysteine, all we have is a carbonyl carbon, alpha carbon, beta carbon. And then that beta carbon has a sulfhydryl group on it, right? So it varies what R group is attached, but they still all have the same pattern. If I take any 2 of these and react them with one another, which is going to be done by translation later on when we get to that lecture on Thursday. So we have a carboxyl group reacting with an amino group, and they lose water between them. One of the H's here and the OH here leaves us water, and it makes the new anhydrous bond. We lost a water molecule, or a condensation reaction, to make what's called our peptide bond, but chemically it's an amide or amide bond. Okay, we saw this once before, and it's how we put our proteins together. Here's our 20 amino acids and their groupings, and I want you to learn all these bases. I think this slide is correct. I think I went through and corrected all the errors in it and some of the drawings that were wrong, like the histidine missing this carbon, right, and the proline having an extra hydrogen somewhere. So there may still be a mistake on it, but I don't think I found any recently. But I want you to memorize these groups, and it's so much simpler to learn them in small groups, like learning your 3 alcohols, learning the 2 acids, learn the 3 bases, learn the 3 aromatics, learn the beta branched ones, right? So it's much simpler to learn them in that way, okay? Which ones are larger than the other? Which ones are the smallest? Which ones are chiral? Which ones are achiral? So put them together in groups, and this will make a lot more sense. Okay, so I want you to learn the names of the molecules, their 1-letter abbreviations, and their drawings. Okay. All right, so let's look back at our amino acid for a second, and let's pick one. Just pick the simplest one. How about glycine there? So glycine, again, has our carboxyl group. It has the amino group, and the carbon number 2, and that's pretty much all it has. Its R group is just another hydrogen. So it always has 1 here, but it's just another hydrogen. So glycine is a rather simple molecule. It has 2 functional groups, a carboxyl group and an amino group. But it'll almost never exist. In fact, it will never exist the way I've drawn it here. Right? This is how we typically draw them when you're learning them, and that's how it was drawn on the previous slides. But this is never going to exist in this form. The reason being is the carboxyl group can deprotonate. It has an ionizable proton, as does the amino group, right? It can gain a proton. It can act as a base. The point at which it does that is called its pKa, right? If you remember back from Gen Chem, pKa is the point at which the 2 species have an equal ratio, right, for protonated and deprotonated. So for your weak acids and weak bases, not for the strong ones, but for the weak acids and bases, like acetic acid, The pKa of that carboxyl group is 4 point something, and I don't want you to memorize it, but that's the point at which half the time it's protonated and half of the population is deprotonated, right? So it's a 50-50 ratio of the two. So if you look at that, in the carboxyl group here on glycine, its pKa is somewhere around 2 to 4, right? Let's just call it 3 for now. And the pKa of the amino group of glycine is somewhere around 8 to 9. Let's just call it 9 to make the math simple here. So if this one's at 3 and this one's at 9, that means if you're at a pH more acidic than 3, this thing will be deprotonated. Sorry. If you're at a pH more acidic than 3, it'll be protonated. If you're at a pH more basic than 3, it'll be deprotonated. For the amino group, if you're more acidic than 9, it'll be protonated. If you're more basic than 9, it'll be deprotonated. So the general theme here is if you're at a higher pH or more basic pH than the pKa, then it's going to be deprotonated. If it's a more acidic pH, a lower number than the pKa, the pH is lower than the pKa, it's in more acidic conditions, it's going to get protonated, right? So this comparison I'm making is always a comparison. We got to get this 7 neutral out of our head, right? If I asked, is pH 7 acidic? Your answer should not be yes, no, or that doesn't make any sense. Your answer should be compared to what, right? So if I asked if 7 is acidic, compared to 4, no, it's quite basic. It's 3 orders of magnitude more basic than 4. So if I said, is 8 an acidic pH? Well, yes, it's quite acidic compared to 12. But compared to 2, no, it's quite basic. So it's always a comparison. So don't think 7 basic and the changes from that being acids or bases. Always think about a comparison. And this goes back to what you learned with your Henderson-Hasselbalch equation. This is exactly the equation that describes the concept I just said. So that equation says if I manipulate the concentrations of the protonated and deprotonated forms, the HA and the A minus, if I manipulate their concentrations and compare it to the pKa of the solution or the pH of the solution, I can determine what form it's going to be in, right? So if the pH is more acidic than the pKa, right, we're at a more lower number on the scale, I expect it to be protonated because it's under acidic conditions. The protons are going to be forced on it. So therefore, the concentration of HA should be higher than the concentration of the A minus form. And the equation sees that out because it's in a ratio and it's a log scale of those. Right? So don't just memorize equations for doing the math. Understand why the equation is in the format it's in, and it's so we can see that changes in these things adjust in a certain way. Okay? So back to our glycine here. We have a carboxyl group whose pKa is, we said, around 3, and the amino group on the left whose pKa is around 9. So if we take glycine and put it in a solution at pH 7.4, which is like our blood solution, right? What would happen to the carboxyl group? So let's make our comparison. pH 7.4 is more what than the pKa of 3?
Acidic.
So pH 7.4 is more what than 3?
Basic.
It's more basic. It's a larger number, right? Significantly larger number, right? It's 7.4 versus 3, right? It's over 4 orders of magnitude greater. Right? It's about 4.5 orders of magnitude greater, right? A little more than that. So we have our COOH there, our carboxyl group, in a pH or an environment that is far more basic than its pKa. Well, at its pKa, it would be 50-50 protonated, deprotonated. But in this far more basic environment, it's going to be mostly deprotonated. The protons will be removed. So at pH 7.4, our carboxyl group will look mostly like this, the COO minus. Right? Same scenario for the amino group. The amino group's pKa is, let's say, around 9. And our pH of the solution, or our blood here, is around 7.4. So in this question, 7.4 is more what than 9?
Acidic.
It's more acidic, right? Not as drastic as the other one was, but it's definitely more than 1 pH unit away, more than tenfold more acidic, right? So our amino group will more likely be protonated in that case, so it's generally written as NH3+. Okay? So this is how I expect glycine to be for the most part. There will be some that are protonated on the end and some that are deprotonated, but for the most part, the vast majority of the molecules will look like this. Right? So that's how we generally write them. And this could have been any amino acid, not just glycine, because they all have these two features. And they all have roughly the same pKa's. But 7 is by far between the two. Right? So our carboxyl group will most certainly be deprotonated, and our amino group will be protonated around the pH of our bodily fluids, right? Like blood. So the Germans came up with a word for this. It's a very simple word, but maybe not so simple if you don't know how to count to 10 in German. But for an ion with 2 charges, you would call it a 2-charge ion, right? And that's exactly what this word is, right? So a 2-charged molecule is a Zwitter ion, right? So zwei in German is 2. So it's a 2-charged molecule. That's exactly what it is. So Germans are really good at coming up with words like this. So it's a 2-charged molecule of glycine. So all your amino acids tend to exist in solution in their Zwitter-ionic form. Right? So they'll have a positive charge on the amino group and a negative charge on the carboxyl group. But overall, it has no net charge. Right? Which one of these two forms, the glycine, which will never exist on the left like that, or the glycine shown in its Zwitter-ionic form on the right, which one is going to be more soluble in water?
On the left.
On the right.
So I heard left and right. What's the rationale for the left one?
No, I meant right, my bad.
You really meant to say right. Okay. What's the rationale for the right one?
It'll readily accept and donate protons with water.
Okay, so will the one on the left if it existed. Right? It can hydrogen bond just as well.
Because of the ions.
Right. Having a full charge can interact with a partial charge on a water molecule better than having just a partial charge on a molecule. Right? So full charges are more soluble than partial charges. Right? So the fact that these two have 2 full charges makes this thing more soluble in water. So that's why glycine exists like this in water solutions around pH 7 to 7.4. Okay? Now what if I change the pH of the solution? What if I take that solution of glycine and make it more basic? Make it very basic. How about we get all the way up to pH 10? Okay? What would happen to this molecule? So what would happen to the carboxyl group? If I made it from you see this the way it looks right now as the Zwitter ion at pH say 7, and I made it all the way to pH 10. I made the solution pH 10. What would change?
You would lose a proton off of the
amino group would get deprotonated?
I'd lose a proton from the amino group, right? Because I've crossed to the other side of its pKa. So it's more likely to be deprotonated. How about the carboxyl group?
They're the same.
Yeah, it's exactly the same. It's already well above, more basic than its pKa. I'm just getting even more from its pKa, more basic. So the percentage that would be in this form would greatly increase, but it still would look mostly like this anyway. So it wouldn't really change. So that's what's shown at the bottom. If I make the solution more basic, right, I'm going to deprotonate the positively charged amino group, and overall the proton or the amino acid will now have a net -1 charge. And that kind of makes sense. As I'm adding base, I'll gain a negative charge, or effectively losing a positive charge. Same effect. Okay? So if the pH is greater than the pi, right, it's going to have a net charge of -1. If the pH is less than the pi, it's going to have a net charge of +1. Well, what is this pi I'm mentioning? Pi stands for isoelectric point. Right? It's not like pH or pKa. It's not a negative log of I. It's isoelectric point. Right? So it's the point at which the ion has, and it does have charges, but it has no net charge. So in the case of our glycine here, that occurs anywhere between, right, the pKa's of the two. So we had one at 6, or sorry, we said the amino group was at what, 9? And we said the carboxyl group was at 3. Right? So Right between the two, around pH 6, would be where we're as far from each of those as we can get. That would be its pI, the average of the two pKa's. Okay? So if I go more basic than that, it's going to start gaining negative charges. If I go much more acidic than that, it'll start gaining positive charges. Okay? And that's shown in a figure at the bottom in one condensed figure. So at its isoelectric point, it has no net charge. If I add base to that, I'm going more basic than the pI. At high pH, or very basic pH, it's going to gain some negative charges or lose positive charges. And the opposite is true at very acidic conditions. You'll gain positive charges from all those protons or lose a negative charge, if you think of it that way. Okay. So let's do a little practice question at the top here. So this is alanine, right? Not glycine, but the methyl group makes no difference here as far as gaining or losing protons. So which structure above, 1 or 2, represents alanine at a pH above or more basic than its pI?
Two.
Right, structure 2, because the neutral one should show alanine with a carboxylate, COO minus, and an NH3 plus, and that structure is not shown at all. But if I get more basic than that, the amino group is going to deprotonate. And that's figure 2. However, if I went more acidic than that, that's question B, alanine at a pH below its pI, more acidic, then we're going to protonate that carboxyl group. Right? So it's going to look more like figure 1. Okay, good. Let's look at that another way using a graph. Right? So in this graph, we're going to show the concentration of 3 different species, the red, blue, and the green one. The blue one in the middle is the zwitterionic form, right, where we have the protonated amino group and the deprotonated carboxyl group with the R group, whatever it might be attached. And then the one on the left in the red is where we've made it more acidic, and we're going to protonate that group, the carboxyl group. Or on the right where it's in green, where we've deprotonated the amino group by making it more basic. Okay? If I look at the concentration of those 3 species, in a solution, the combination, the sum of all 3 concentrations will be constant because it has to exist in one of them. So if I add up the concentration of the red form, the blue form, and the green form, I get a constant. But in some ranges of this chart, one of them will be virtually 0, right? So if I was at pH 1, right, if you look on the graph, some of it exists in the zwitterionic form, but not very much. The majority of it will exist in the red form, the protonated form. But there'll be almost 0 of it in the green, doubly deprotonated form. Right? There will be a few molecules in there of the bajillion molecules that are in the solution that will be in that form, but it's a by far minor contribution to the number of molecules, or in that total sum. As I get more basic, you notice that they switch places. More of it takes on the zwitterionic form and less of it the protonated form, and where the lines cross, where the red and blue lines cross, at that point, the concentrations of red and blue are the same. That's the definition of pKa. That's where the concentration of the 2 is equally likely. So where the red and blue lines cross, let's say that's what, pH 2 in this example, right, according to the graph, that's the pKa of what group?
Carboxyl.
Of the carboxyl group, right. The carboxylic acid's proton has a pKa of 2 in this example. What would be the position where the blue and green lines cross? What would that represent? So the blue and green lines having the exact same value means I have a concentration equivalent between the two. What was that? Sorry, I couldn't hear you. So where the blue and green lines cross, the concentrations of the blue and green species above would be the same, and that would be where the pKa of whatever change occurs. And the change there is where the amino group gains or loses its proton. So where they cross around 9 or so, right, like 9.1, 9.2, somewhere around there, is the pKa of the amino group. Okay. So which amino acids can do this? Right? So we know there's the amino group and the carboxyl group on the ends, right, which are listed here in the first and the fourth line in this chart. So at the very top of the chart, we have the carboxyl group called the alpha carboxyl group because it's on the alpha carbon. It's carbon number 1 on the alpha or 2 carbon. It has a typical pKa somewhere between 2 and 4. Right? I don't need you to memorize exactly 3.1 because it does vary, but on the acidic end, somewhere between 2 and 4. Right? And then the terminal amino group, the fourth line in this chart, has a pKa somewhere around 8 to 9. Right? You don't have to memorize exactly 8.0, but somewhere between 8 and 9 on the basic side. Right? And then if you look, all the others are your acids and bases and a couple others in this group. So your 2 acidic amino acids, aspartic and glutamic acid, have a pKa much like the carboxyl group at the end of the chain, around 2 to 4 again. Right? So they're also carboxylic acids. That makes sense. Your histidine is an amine. It's one of our bases, but its pKa is around 6. Okay? That doesn't, just because it's 6, remember you got to get that 7 thing out of your head. So just because it's 6 doesn't mean it can't be a base. And the reason we call it a base is because upon gaining a proton, it gains a positive charge. Right? The reason we call aspartic acid an acid is because upon losing a proton, it adopts a negative charge. That's what acids do. Right? HA and A minus. Whereas bases become B and BH plus. Right? So that's our definition for that. So histidine's acting as a basic amino acid, but its pKa happens to be 6. Right? The other 2 bases are at the bottom of the chart, the lysines and arginines. So lysine's pKa is around 11, and arginine closer to 12 to 13. So it's the most basic one. And again, it's because their amino groups are gaining protons and they gain a positive charge. We're going to call them basic amino acids. Right, and the only 2 left in the list now are some unusual ones that aren't truly acids or bases in the sense of what we just said, but they're kind of unusual creatures, right? So normally alcohols, the pKa of most alcohol groups is somewhere around 16. You know, like ethanol or a serine or a threonine amino acid. The pKa's are well above that, around 16 or so. So it's unlikely to lose its proton. It's a terrible acid, right? But tyrosine, that's a phenolic group, right? So as you remember from your organic days, phenols are slightly acidic, right? So they have a pKa that's around 10 or 11, right? So tyrosine mirrors that, and tyrosine somewhere around 10 or 11, right? Much like lysine. Because the negative charge that develops from the extra lone pair after deprotonation can be destabilized by or stabilized or delocalized into the benzene ring. Right? And the last one on the list is cysteine. Right? So cysteine, again, like an alcohol, normal alcohols won't be able to do this, but cysteine, instead of it being a normal alcohol, is a thiol, and that lowers the pKa from around 16 to much closer to around 8 or 8.3. Okay, so memorize these numbers in their relative ranges. You don't have to memorize exactly 3.1 or 12.5, but get a relative scale in your head of what these look like. Okay, we're going to refer to them quite a lot as we talk about other things later. Most of the other amino acids in this list, like methionines or leucines or isoleucine or valine or all these others hydrophobic ones, really don't have a pKa or a deprotonatable or protonatable group to speak of. Right? Some can be talked into it, but for the most part, these are the ones I want you to know. Okay? So the common ones. All right, let's look at an example for alanine again. So on the far right here, I have alanine drawn in 3 different forms. So in the middle is our zwitterionic form. You notice it's got that carboxyl group with the negative charge, and the amino group with the positive charge, and of course its R group is just a methyl, which does absolutely nothing. Right? So we can make it more acidic. That's above it. It's called the anionic form, right? Because the overall charge is -1. Yes, it did gain a positive charge by doing this, right? In the anionic form, it gained a positive charge, right? But it's called the anionic form because the overall net charge is -1. Whereas if I go to the bottom, negative charge. If I go to the bottom and make the cationic form, it gained a positive charge. And now it has an overall +1. So of these 3, the top one, which is a -1, the bottom, which is +1, and the middle, which is no net charge, we're going to call the middle one the zwitterionic form because the net charge is 0. Okay? So where that molecule is predominant, we're going to call that the PI, the isoelectric point, where the no net charge molecule is the most abundant type. So how do we figure that out? Well, I need the pKa's of the two groups involved. The carboxyl group has a pKa of 2.4. We said it'd be somewhere around 2 to 4. This one happens to be 2.4. It varies from amino acid to amino acid. And the amino group here happens to be 9.9, right? It could be 8, 9. This one happens to be 9.9. So it's a little more basic than we expect for, say, glycine. So where do we find out the isoelectric point? We said we take the two pKa's and average them. So if you take 2.4 plus 9.9 divided by 2, the result will be the pKa somewhere around 6, right? So just above 6. So if you look at our graph here, what we're graphing is the pH of a solution with alanine in it here, and I keep adding base. Well, the alanine will do this too, but alanine is acting as a buffer, right? It's resisting the change in pH. So if you follow the curve, as I add hydroxide, if alanine was not there, this would just go up linearly, right? As I add OH, the pH would just keep climbing, right? But it's resisting the climb. You see it's curving over, right? There's an inflection point here, right? It goes from a positive curvature to a negative curvature, right? But at some point, it can no longer resist and it goes up quickly again, right? The curve starts going up rapidly again. But then we approach another region where it's starting to resist the change in pH one more time, and it curves over. Again, yet another inflection point, right? And then it goes up again after that second pKa, the pK2. But in the very center, we have another inflection point, right? Where it changes from curvature positive to negative one more time. And that's where we're transitioning from one side of the PI to the other side of the PI, right? So this inflection point, which is the average of the two pKa's, is the PI. Why are there two flattened points here? Because there's two groups that can gain or lose that proton. So as I'm adding my hydroxides, my strong base, the weak acids and bases are compensating for the change. That's why you get these curvatures, right? Why do we see two curvatures? Because there's two functional groups that can gain or lose the proton. Okay? So the isoelectric point is pretty easy to calculate. If you have two pKa's, you just take the average of the two. Right, so here's a question for you. How can you have the same amino acid, let's say histidine, for instance, which you know its pKa should be around 6.0. So how can you have histidine have two different pKa values when that same amino acid occurs in different parts of the same protein? So maybe histidine in one area of the protein and another histidine in another area of the protein aren't necessarily the same pKa. How is that possible?
Be due to the region of the cell that the protein is in. So if we're thinking about like a transmembrane protein, for example, where you have part of it in the aqueous cytosol and part of it in the cellular membrane.
So that's an extreme example of the answer I was looking for, but think on a smaller scale. You're absolutely right, but think on a smaller scale. Not necessarily so much where it is in the cell, but the environment in its immediate vicinity. Okay, so if I had a histidine that's in a region of the protein that's surrounded by a bunch of aspartic acids. Right? So there's a bunch of aspartic and glutamic acids nearby, near my histidine. Do you think the histidine's pKa, and think about this before you answer, do you think the histidine's pKa will be 6.0 or more acidic or lower than 6.0 or much higher than 6.0? So you have a histidine surrounded by a bunch of aspartic acids in that local microenvironment. And if the histidine were all by itself, we'll say its pKa is 6.0, if it were just isolated. But in this microenvironment, what do you think will happen to its pKa?
Become more acidic.
It's going to be more... I didn't hear what you said.
More acidic?
More acidic. It's more likely to lose its proton. Is that what you're saying?
Or no, it won't accept protons more because the acids are donating their protons at a
The
higher
acids exactly will readily donate their proton to this histidine. So from an outside observer, is it more difficult to remove the proton from the histidine or easier?
It'd difficult.
More difficult. What does that mean about its pKa?
It's got to be higher than 6.
It's going to be higher than 6. It's more difficult. I'm going to have to add more base to get that proton removed, because the surrounding amino acids are more likely to put it back on, right? Now, what if it were surrounded by a bunch of arginines and lysines? So now what will the pKa of that histidine be? So if I surround it by arginines, and the pKa won't be 6, but closer to what?
It'll be more acidic.
More acidic, more like 5 or 5.5 or something like that. It'll be on towards the lower scale, because those arginines will keep stripping the proton away, right? It's very difficult to remove it from the arginine because their pKa is like 12, 12.5. So every time I try to protonate this histidine, right, by dropping the pH, they're going to keep taking it away from it. So it's harder to put the proton back on, right? So its pKa will be much lower. Okay, it's more willing to get rid of it because there's someone accepting it nearby. Okay, so another question for you. Under physiological conditions, which means pH 7.4, an amino acid like glycine has a net charge of what? So think about the structure of glycine. It's very simple. It's R group's just a hydrogen.
It's neutral charge.
So it'll have no net charge, 0, right? And why?
Because it's in its ion form, you're going to have the positive charge on the amino side and the negative charge on the carboxyl side. So
Right. So
It'll be in this Vitter-ion form.
right. It'll be in its Vitter-ionic form, where like you said, the amino group will be positively charged because the pH is more acidic than its pKa, and the carboxyl group will be negatively charged. It's lost its proton. It's been deprotonated because the pH is more basic than its pKa. So the same pH can both be acidic and basic. It depends on what you're comparing it to. That's the idea I want you to get. Okay. Each amino acid has at least 2 pKa's. Explain why that's true.
Two places. Go ahead.
That would just be the 2 functional groups. You got at least the carboxyl group and then the R group, whatever that side chain is going to be.
Well, not necessarily the R group, but it has, you're right, the carboxyl group
I'm sorry, the amino group.
and the amino group, yes. And the R group, we're going to ignore it for now because it says each one, so they vary. So each amino acid has at least that. I agree. Okay. So what does it mean if I'm asking the same thing multiple different ways here because I want you to understand the concept. So what does it mean if the pKa of an amino acid's R group is 7? Please don't say neutral.
That it can behave as an acid or a base depending on the environment it's in.
No. What does it mean if the pKa of the amino acid's R group is 7? Think about what pKa means. This is literally the Henderson-Hasselbalch equation in written form.
Both of them are the same amount in solution.
What do you mean by both of them? No pronouns.
Equal amounts of acid and base.
So the R group can be protonated or deprotonated, and the protonated form and the deprotonated form exist in equal concentrations. That's what you were saying, right?
Yeah, in worse terms.
That's okay. It's the concept you get across. So you can put it in your own words. So the R group, it varies among amino acids here. If I say it has a pKa of 7, it means that group can exist in a protonated and deprotonated form. And when those 2 concentrations are equal, right, that's its pKa. Well, that means if its pKa is 7, those 2 concentrations will be equal when the pH is 7. Because if you look back at the Henderson-Hasselbalch equation, remember it's pH equals pKa plus the log of the ratio of those concentrations. If the pH equals the pKa, that latter term must be 0. And if the latter term is 0, the log of something is 0, that something is 1, and the something is the ratio of the two concentrations. If the ratio of the two concentrations is 1, they're equal to each other. So that's the definition of pKa again. So that's what that equation really says. That when the molecule exists in a pH that's equal to its pKa of that group, then that group exists in equally abundant protonated, deprotonated forms. That's all this question's asking. Okay? Histidine has 3 pKa's. Tell me why. We said they all have at least 2. Why does it have 3?
It has a charge on an amino group in the R group.
Right, so the R group is, like we just said in the previous question, a deprotonatable protonatable group, right? So in histidine, it happens to be an amino type group. So it's an imidazole ring, but we can think of it as another amino type group. And so in that case, it can gain or lose a proton. So there's another pKa for the R group. So it has 3 pKa. So it's carboxyl group, it's amino group, which all amino acids have. And then there's the imidazole ring. That's the ring of the histidine with those 2 nitrogens in it. That side chain of histidine can be protonated or deprotonated. So let's look at that one like we did our alanine. Okay, so here's the same analysis that we did for alanine, except this time for histidine. So in this case, instead of having 3 forms, it can exist in 4 forms. So looking at the bottom part of the picture where the molecules are going back and forth, the one on the left, you notice the carboxyl group here is protonated, the amino group is protonated, and the imidazole group is protonated. So all of them have their protons. This is under very acidic conditions. Okay? As we go past the first pKa, which is the one for the carboxyl group, right, which is somewhere around, right, remember we said carboxyl groups around 2 to 4? This one happens to be a little more acidic than that. It's 1.82. But then this one loses its proton, right? At 1.84, this top molecule, or this top group, would exist 50-50 between these 2 species. But as we go more basic than 1.82, it starts to look like this. Okay? And then as I keep going, and I add more base, eventually the next pKa I come to will be the imidazole ring, which is around 6.0. Right? So this nitrogen here will lose its proton. It'll be 50-50 protonated, deprotonated at 6.0. But as I go more basic than 6, it tends to look like this. Okay? If I keep going, finally the amino group will lose its proton, right, around 9 or so. This one's 9.17. So these two, 50-50 at 9.17. But eventually, if I go more basic than that, it looks like the molecule on the far right. And you see that we have those 3 plateaus as we go up this titration curve. Okay? Well, now I have a problem. If we look back at the last one, our PI was the average of 2 pKa's. It was right between the 2. So what do we do now? We have 3 pKa's. How do we find the PI? Do we average all 3? How do I solve this problem? Have any ideas?
Would there be 2 different PIs?
There aren't 2 different PIs. Let's think about what the definition of PI is one more time. PI is the isoelectric point. It's the pH at which what occurs?
The Zwitter ion and whichever form of the molecule you're looking at are in equal amounts.
So the molecule, what you just said, but the molecule has no net charge. So it'll be in a Zwitter ionic form, but it has no net charge, right? So which one of these 4 species I have drawn here has no net charge? The first, second, third, or fourth one? Well, the first one, let's look at it. The carboxyl group is neutral, has no charge. And you have 2 positive charges. So this one on the far left has a +2 charge. And if we draw these correctly, we should keep dropping 1 positive charge or gaining a negative charge every time we deprotonate something. So this one has a +2 charge. This one has a +1 charge because 2 positives and a negative, we have a +1. The third figure has a neutral ring, right, and a positive and a negative here. This has no net charge. And the last one has just a -1 charge. So the third figure, right, is our one with no net charge. Where does that occur in our graph here? Well, that's between the pKa's for the R group and the amino group. So between 6.0 and 9.17. So this region is where the pi is, the neutral species with no net charge. So I would average 6.0 and 9.17 to get this point here just below 8, right, 7 point something, as our pi. That's possible as long as the other pKa is significantly far away from this one. If the first pKa happened to be like 5.7 and 6, then it would have an effect on this. But it's so many units away, it's virtually non-relevant for finding the pi here. Because even with this one with no net charge, if I go more negative or more positive from it, the carboxyl group remains exactly the same. Its concentration to the protonated form really doesn't change this far away from its pKa. So if you have one that has an R group that can be protonated or deprotonated, I gave you a very short list to learn, then there will be 3 pKa's. And you got to know which two to use. It's not always these two, right? You got to know which two to use to calculate the pi. It'll be these two for the basic ones like histidine, lysine, and arginine. And it'll be these two for the acidic ones, right? Because we're going to generate a negative charge from that R group, not a positive charge for these. So find the two that surround the neutral species. We did that. And then average the two numbers, assuming the other one is significantly far away, and they generally are. Okay, so make sure you can do that. Okay, so let's look at our amino acids real quick. We'll go through a rundown of the 20. I do want you to learn them, but they do have some peculiarities to a few of them, right? So I say here that 19 of our 20 amino acids are chiral, right? They're enantiomers, right? Enantiomers exist of the 19 of the 20. Which one is not?
Glycine.
Glycine, good. So to be an enantiomer, to exist as chiral molecules, you have to have a chiral center, right? So our alpha carbon here is in a chiral center as long as the R group doesn't match one of the other 3 constituents, right? So in our case of glycine, the R group is another hydrogen, and therefore all 4 things are not different. So glycine is achiral, or not chiral. The other 19, however, the R group matches none of the other components, so they're all chiral. And for every protein we make, we only use the L isomers, right? The levo isomers, not the dextro isomers. So all your proteins in your body and all proteins in every organism are made of L isomers exclusively. Now, I have to say this because someone's going to say that this isn't true. There are D isomers of amino acids in some proteins, and that's an after-the-fact addition, right? So we add some D isomers. This happens in, say, the bacterial cell wall, right? There are some D alanines in there, but they weren't made that way at the ribosome, right? They were added later, right, or changed later. So they were post-translationally modified. So all the amino acids that the ribosome uses to assemble this thing are going to be L isomers, okay? So keep that in mind. You'll see a commonality to that when we get to our sugars later, that we always use the D isomers of our carbohydrates, like our ribose. It's always a D isomer, whereas all the amino acids are the L isomers, because those two things bind one another very well. Right? So that's the reason for that. Had we chosen to use D isomers of amino acids and the L isomers of our carbohydrates, that system would have worked just as well. Right? But that's not what happened. So we tend to use the L's for amino acids and D's for sugars. And another peculiarity of the list is our proline. Proline is the only one in which the R chain is just a 3-carbon a propyl group, right? Just 3 carbons sticking out, actually wraps around and attaches covalently to its own amino group. Okay, it's the only one that does this. Don't confuse this with histidine. Histidine does not do this. The R group contains the ring exclusively. Here, a ring is formed because the R group makes a connection to its own amino group. Okay? So proline is the only one that does this, and that puts a little constraint on this thing. The carbon, alpha carbon to amino nitrogen bond can no longer rotate. Right? So the carbon nitrogen bond could normally rotate, right? But now because of this ring bonds don't rotate. So it puts some constraints on the geometry that you can adopt with a proline. Okay. Some other unusual ones, or stands out from the list, are the two that contain sulfur. Right? So methionine, which is, the name gives away what it is. It's a methyl thio group, right? Like a methoxy ether group, but instead of an oxygen, it's a sulfur. So we call it a methyl thio. So methionine, the name gives away what it looks like. And the other one is cysteine, which I'm mispronouncing, much like everyone does, universally mispronounced. I don't think I've ever heard anyone call it correctly. We call it cysteine. The actual correct pronunciation should be cysteine with 3 syllables, but no one does it, right? We pronounce it cysteine. But cysteine can do something very unusual that only cysteine can do. No other amino acid can do this. Is 2 cysteines can come together and form a disulfide bond. So each of the cysteines loses a proton, right? Loses a hydrogen, and one sulfur bonds to the other. Right? We make a sulfur-sulfur bridge or a disulfide bond. Right? Is there another amino acid very, very similar to cysteine that might be able to do this? Well, if we remove the S and replace it with an O in cysteine, what amino acid would that be?
Serine.
It would be serine, right. So could 2 serines do this? Because sulfur and oxygen sort of behave similarly. Could I make an oxygen-oxygen bond across 2 cysteines? 2 serines?
Wouldn't that just be a peroxide?
That's exactly right. And that's why it generally does not form. It's very unstable, and that would be a peroxide. Peroxides are not very stable, so it tends to not happen. However, the disulfide bridge, or disulfide bonds, are very stable. Right? So much, much more stable than the peroxide situation. So cysteines tend to do this under oxidizing conditions. Under reducing conditions, they're more likely to come apart into 2 sulfhydryls. Okay? So back to the spelling and the pronunciation thing. We generally call the amino acids cysteine. And when they come together, they form that formation. You see it spelled differently. And we're so ingrained to mispronouncing cysteine as cysteine, because that should be pronounced cysteine now. We're so bad at mispronouncing it, we call this cysteine, because we've mispronounced the other one so badly, right? And it's caught on so much, that's the only way we say it now. So it should be pronounced cysteine as the individual and cysteine as the disulfide, but no one does that because we mispronounced the original. So we call it cysteine, which sounds awful, but that's what we pronounce it, and it's stuck, right? So I want to ask you to make the difference in the two, but know that cysteine is the only one that can do this, and methionine cannot. So looking at the structure of methionine, why can't we form a disulfide bond with methionine?
The CH3 in the way?
Sorry, I couldn't hear you.
The CH3 in the way?
Yeah, there's a methyl group in the way. So in order for cysteine to do it, all I need to do is deprotonate it, right? Remove it. And we already know its pKa is about 8.3. So it's not unreasonable to say, even at pH 7.4, that roughly 10% or so of the cysteines are going to be deprotonated, because we're only about 1 pH unit away from its pKa. However, methionine is very, very unlikely to lose a methyl group and leave a negatively charged sulfur behind. So the sulfur kind of precludes the formation of disulfide bonds. Sorry, the methyl precludes formation of disulfide bonds with methionine. So only cysteine can do this. Okay? So a really short story to date me, to tell you how old I am. When I was in high school back in the 90s, and you went to prom, and you know, it was one of those things, you got a perm in your hair. I regret it, but I'm not embarrassed to tell you I did it. My hair is perfectly straight, no curls, no curves whatsoever. So when you go get a perm in your hair, and many of you probably don't do this anymore. It's a 80s and 90s thing to do, although it still happens and people do it. But you may not understand how it works or have seen it done. When you go get a perm put in your hair, yes, I know you're laughing at me now. But when you go and they add some chemicals to your hair, and what it's doing is making and breaking these disulfide bonds. So if you have really curly hair, right, there's a lot of disulfide bonds holding it into that shape. If you have less curly hair, there are fewer disulfide bonds. And if your hair is perfectly straight, you have very few at all, right? So I was going to get my hair, permanently, semi-permanently put into a certain wave or feature or whatever it was at the time. I don't remember. But what they do is they add an agent that will break all the disulfide bonds. So that's going to be a reducing agent, right? It doesn't smell so good. So most reducing agents don't smell great. Neither do oxidizing agents, but reducing agents really smell bad. And it breaks all these disulfide bonds. They put your hair proteins in the shape you want them in, rinse them out, and then you add an oxidizing agent. And it'll put those cysteines and it'll reattach them to their nearest other cysteine neighbors. Maybe not the best stabilization, but it's called a perm, but it's not so permanent. It's a temporary, we should call it, right? And so when you leave the salon, the first thing you should not do is what? Maybe some of you know.
Don't wash your hair.
Don't wash your hair. Don't get it wet, right? Because water will break a lot of these bonds and then allow them to relax into a more natural or stabilized formation, and then they'll reform there. So you'll lose all the work just put into that nasty reduction oxidation process. And that's how the perms work, right? So they're not permanent. They should be called temporaries from now on. Okay. Right, so our last thing I want to talk about is our amino acids broken down into two categories. So we'll get to the fella at the bottom in just a second. So look at the top of the screen. We have our 20 amino acids listed as non-essential and essential. And virtually everyone ever asked will agree on 19 of the 20. Okay? The one everyone disagrees on is tyrosine. Okay? And I also agree that they're both right. So should tyrosine be put in the non-essential category? Yes, I agree. Should tyrosine be put in the essential category? I also agree with that. The reason is the definition behind the word essential, right? That's what makes the distinction. So the other 19, everyone agrees with, right? So if you're writing a book, like this table came out of a book, and they put tyrosine in the non-essential column, they must have a definition for a non-essential that says you can make it from something else. That's what non-essential means to them. Okay? If you put it in the right-hand column, it means you cannot make it from anything else. You must get it in your diet. That's what essential means. Okay? And I agree with that definition too. If you're putting tyrosine in the essential column on the right because you want 2 columns of 10, that's a bad idea. That's not a valid reason. Okay? Some people do that, right? Just because they like the look of 2 columns of 10. But if your tyrosine, if your definition is you cannot make it from anything else, right? You're going to put it in the essential column. So this book clearly goes by the definition you can't make it from scratch, but you can make it from other things, right? And in fact, you can make tyrosine from phenylalanine. We just hydroxylated, right? So this book agrees with tyrosine being non-essential with the caveat that you're being supplied with phenylalanine because it is essential. So if you're not being supplied with phenylalanine, you're also not making tyrosine. So that's why some people will like to put it in the essential column either way. I agree with both arguments. It's just how you define your definition of essential is what matters. So that's why you might see it in another column, in another situation or book or a table somewhere. Okay? So let's get to the guy at the bottom. Does this guy look like he's well fed? The little fella on the right, does he look like he's well fed?
No.
He's got a really distended abdomen. He is getting food, but there's something missing from his food, right? He's getting lots of some things and very little of other things. So the thing he's missing in his diet is protein, right? Probably getting plenty of carbs, lots of lipids. You can't avoid nucleic acids in your food unless you're not eating at all. If you're licking rocks, you're getting nucleic acid because there's bacteria there, right? But he's not getting enough protein in his diet. So what's happening? Why do we see the protruding abdomen, right? So what's happening is if you don't eat enough protein in your diet, your blood becomes a lower concentration of amino acids. Okay? So now we're going to dredge up some old terms from high school biology or wherever you took this class. So if your blood concentration has decreased in amino acids and the tissues around it have not, The blood is what compared to the tissues? I'll give you a hint. It ends in the phrase tonic.
Hypertonic. Hypotonic?
Hypotonic, right? There's fewer things dissolved in the blood, the amino acids, compared to the tissues around it, which still have a lot of amino acids in them. So initially, when you're not getting protein in your diet, it doesn't end up in the blood, your blood becomes hypotonic. What happens to a hypotonic solution when it's surrounded by a more hypertonic solution?
It shrinks.
It's going to lose water, right? The proteins, amino acids outside, can't leach into the blood because they're attached to other proteins, right? The amino acids. But the water can leave the blood and go into the interstitial fluid, right? Into the tissues around it. So the blood volume will decrease to try to make it isotonic with the tissues around it. So the blood volume will decrease, right? And the blood, or not the blood, but the tissues will become more hydrated around it. And of course, if you do that, and you do that for a very long time, eventually all that extra fluid is going to pool in the abdomen, right? It's the biggest chamber that it can pool in. It can't pool in the skull because your skull generally doesn't expand, but your abdomen can, right? And it won't pool in the muscles so much because they're dense protein, but it'll pool in the abdomen because you have a big cavity and gravity will pull it down to there rather than the chest. So he has this huge distended abdomen because of all the collected fluid, right? So if you catch this early, you can fix it, right? But all this is lowering his blood volume. He's going to have a lower blood pressure, so the heart's going to have to work harder to pump the fluid, right? And so this eventually could kill someone if you go too long. So if you came across this little guy while you were hiking, what would you do to fix it?
You give him some beef jerky.
Okay, you just killed him.
Okay, don't do that.
Yeah, don't give him a beef jerky, or your protein bar or anything. That you just killed this guy. So what would happen if you don't worry? Everybody says that. I'm not picking on you. What would happen is suddenly there's this rush of protein, right, that you're eating. It's going to be broken down to amino acids in the gut, and then there's this influx of amino acids into the bloodstream, right? So now the blood becomes what compared to the tissues? Quickly.
Hypertonic?
Hypertonic. Where's all the fluid going to go?
To the blood.
That's our goal, but not this quickly. All that fluid is going to rush back into the bloodstream, and what's going to happen to the blood pressure?
It's going to skyrocket.
Skyrocket. You're probably going to blow an aneurysm in the skull or something, or you're going to have bleeding and lots of capillaries. And what's going to happen to all the other metabolites in the blood, like the sodium and the other ions there?
Really diluted.
You're going to dilute them out so much that you're going to interrupt electrical conductivity and you're going to stop his heart, right? Because you're going to run out of ions. So let's not give him the beef jerky. Let's go about this a little more carefully. What would you give him instead? Well, here's what I would do, and I always use this example. Assuming I know he's not allergic to peanuts, I give him a peanut. All right, eat that. Come back in 10 minutes. I'll give you 2 more. All right, go eat those. Come back in a half hour. I'll give you 3 more. Slowly build up the amino acids that are going into his system, so that this influx of fluid happens more slowly. Right, and then you won't have him have a heart attack and an aneurysm burst and everything else. All right, so slowly get the fluid back into the body, and it won't have this detrimental effect of killing him. All right, last slide. I want you to look at a few amino acid derivatives, right? So these are based on our 20 amino acids, but they've been highly modified. In some cases, not so much, but some cases very modified. So what happens is the protein is made, it's assembled, and then we're going to take these extracellular or even sometimes intracellular proteins and modify the protein, right? We're going to put groups on it, like we did with our RNA, putting methyl groups and making it pseudouridines and doing some modifications. So can you figure out which amino acids these started out life as and they've been modified? So some are fairly easy, they're in the name. So looking at the bottom left here, right, so of the figure, we have 4-hydroxyproline. Can you tell that was originally a proline, because it's in the name of course, but it's been hydroxylated. So at the which carbon am I putting the OH? What number carbon is that of the proline that receives this new red hydroxy group?
4.
Number 4, or in the Greek system, it's the
Delta.
not delta, gamma. Remember the carboxyl one doesn't get a letter. All right, so this is the carboxyl group, then the alpha, beta, gamma. Remember the amino group is on the alpha carbon. And so on the gamma carbon, I put an OH. So it could say 4-hydroxyproline or gamma hydroxyproline. And what's the point of putting this on there? If you don't do that step, your cables that hold your body together, you have some proteins that act as fibrous proteins, which will come next time, but you have some cables that hold your body together, and they're made of a lot of these prolines, right? And if you don't hydroxylate it, you can't attach the cables to anything. They're going to be very hydrophobic proteins, right? It's just proline and glycine. That's all it is, right? So if I don't attach that to anything, the cable doesn't do any good. So I need some way to interact with the protein. So these hydroxyl groups are ways that you can anchor these cables, like to your muscles and to your bones, to make these strong proteins. It will cover what these cables are later, right? But I want you to understand that you have to modify some proteins here. And if you can't modify it, then you have things like scurvy, where you don't connect things properly, right? So I need that hydroxylation event, or you're going to get something called scurvy. Okay? A similar event underneath, we have a hydroxylysine. So in this case, we've hydroxylated the R chain on the lysine near the end. And the one underneath it, I've taken a lysine, and instead of hydroxylating it, I methylated it. This is very similar to what we did in our histone proteins, right? Instead of methylation, we did a acetylation, where we put an acetate on. Here we put a methyl group on. So another modification. At the top center, we have a carboxyglutamate. So we've taken an already acidic R group, a glutamate's R group, and added yet another carboxyl group. So this might make it much more soluble in water. It's going to make it much more interactive with things. I can connect things. It's branched now, right? On the far right, I have what's called alysine, right? Where I've taken a lysine and replaced the amino group on the end with a carbonyl, right? So the very end used to be an amino group, and I've replaced it with a carbonyl, and that carbonyl is in the form of an aldehyde. So that's why it's called al-lysine. It's an aldehyde lysine. So this lysine with its aldehyde functional groups, and you may remember from organic that aldehyde groups are very reactive, could be used to cross-link proteins. So I can cross-link this protein with one nearby, and now they're covalently linked. Okay, that's one way to do that. At the very bottom of the picture in the center is called selenocysteine. This is considered the 21st amino acid because it's like the serine, where there's an oxygen there, and it's like cysteine, where there's a sulfur there. If I go right down the periodic table, under oxygen, under sulfur, under there is selenocysteine, right, or selenium. So I put a selenium in place of the sulfur, and now I have selenocysteine. And it's right on the border of being a metal and non-metal, so it has some unique properties used by some enzymes. And you have selenoenzymes that you use as well. Okay? The tough one to see is the center one. Can you guess what amino acid desmosine is made from? And in fact, it's made from 4 identical amino acids. It's 4 of the same amino acid put together to make this complex. Kind of highlighted them for you. Any guesses on what amino acid appears 4 times there?
Lysine?
You say glycine?
Lysine.
Oh, lysine. Yes, that's absolutely right. It's lysine. So if you look on the top one, you'll see that it's got the normal carboxyl group, amino group, shown in the zwitterionic form, of course. And then lysine has 4 CH2s followed by an amino group. So this one, you see there's 1, 2, 3, 4, and the amino group has been lost. Okay. The one on the right, we again have our carboxyl and amino groups, and then we have 1, 2, 3, 4, right? The amino group has been lost. The one on the left, again, here's your alpha carbon, and then 1, 2, 3, 4 carbons, the amino group's been lost. And the one at the bottom, I have all the parts, the carboxyl amino group, alpha carbon, and then 1, 2, 3, 4, and its amino group. So through the loss of 3 amino groups, I can form this ring structure in the center that holds all four of these together. What's the point of something like this? Much like our L-lysine on the right, it's used for cross-linking things, or taking multiple protein chains and have them all hooked together. Right? So these four chains might be held together by this point. It's a way of keeping subunits together permanently. Okay? So keep in mind, our amino acids, once they're made into proteins, they're not done. They can be modified afterwards. And I'm teaching a whole class on that in the fall of post-translational modifications, but you don't have to take it. But many, many proteins, amino acids, are modified after their birth at the ribosome.